Okay. Hello, everyone. My name is Noeen, and I am Director of Communication and Events at SCWIST. Thanks for joining us today for our second last session for Symposium. Um, Before we go ahead, I would like to give land acknowledgements. We would like to take a moment and reflect on our connection to the land and thank the traditional guardians of that land which we at Squeezed Vancouver live and work. Vancouver is on the unceded ancestral and traditional lands of Musqueam, Tsleil-Waututh, Seychelles, Squamish, and the indigenous people of Turtle Island. Today is second session of round 2 for category global warming, earth and environmental science. There are some housekeeping rules. Please note that today's event will be video recorded and it will be uploaded to our YouTube channel later on. We will take out that we will keep our mobiles and also our microphone muted. Chat box will be monitored and questions and answers will be taken during our question and answer session for audience. I would like to introduce our sponsors who are sponsoring this symposium and bringing this wonderful event for our students. Our first sponsor is Northeastern University Vancouver. They have very highest employment rate for biotechnology. If your field is biotechnology or related to computer science, you can definitely look for more programs on their website. Our another sponsor is Admire Bioinnovations. If you would like to polish your skills in business or science, check out our programs and different programs related to business skills and also related to science experience at Admire Bioinnovations Academy. For Epsilra, please visit the Epsilra website for their recently discovered antibody that neutralizes viral variants of COVID-19. For Microsoft, you can check their website for internships in information and technology. Equitas Therapeutics, it's a very small startup company based in UBC. They have, and it is really accelerating very fast. They provided their lipid nanoparticle delivery system, a key element in the development of Pfizer's vaccine. Kruger joined us recently. They developed a revolutionary bio-based material with unique properties, which is called as phytocell. This new additive will enhance the performance of your products, unlike any other, and can be leveraged in numerous applications, including automotive, construction, aerospace, to name a few. In addition to that, we have other sponsors also like Sophos, which is for cybersecurity, Six and Strategy Group, which is for life science consultancy group, Molly Surgical, which provide and works on research related to cancer, like especially breast cancer. We have two wonderful judges today with us. One is Claris Canta. She did her master's in science from Australia, and she is working as internal systems coordinator, student Energy Canada. And Cecilia Sierra, she is MSc MA from UBC. She is a lecturer and faculty of health sciences at SFU Canada. We have our speaker with us today, who is Kristen Hayward from Queen's University, Canada. She will be presenting about a new genomics method for non-invasively monitoring Canada's polar bears population. From here, I will stop sharing my screen and I will give the floor to our judges to introduce themselves and then to Kristen Hayward. Thanks everyone for joining us today.
I can start. Thank you, Noeen, for this generous introduction. I'm just going to add that I'm lecturing in 2 courses at SFU in research methods in health sciences and environmental health, how the environment intersects with the human health and everything that surrounds us. And I'm extremely privileged from this series and I enjoy learning from all of you. So looking forward to your presentation, Kristen. Thank you.
Yeah. Hi, Kristen. Hi, everyone. My name is Claris, and I'm currently affiliated with Student Energy. It's a global youth-led nonprofit. We're focused on empowering young people who are leading our transition to just and sustainable energy future. Before moving to Canada, I'm actually working in project development of renewable energy projects, specifically solar, wind, and geothermal. I started my career as a geologist, but then I realized I want to learn the vast energy field. So I decided to take my master's in energy systems in University of Melbourne to expand my knowledge on technical and also the business aspect of energy. My personal interest is definitely in clean energy transition and the climate and energy nexus. Yeah, so with this event, I'm really learning about a lot. Like global change is such a massive, massive field, and it's very interesting to hear all your research and studies. So I'm looking forward to that. Pass over to Kristen.
Hi, everyone. Thank you for having me here today to speak. I am Kristen. I am doing my master's at Queen's University. I'm just finishing up my second year, probably going to go a couple more months here, but I'm studying under Dr. Stephen C. Lougheed, who is here today as well. I did my undergrad at Queen's as well. I did it in a biology and psychology specialization, which was really cool. And in my fourth year, I decided to do a thesis very last minute with Stephen and got into polar bears. So I've been studying them since about 2018 now. It's a passion project. I'm really, really excited about it. And kind of as an aside, I have a bit of a side project with Andean condors as well. I'm working on a nest for that. So I'm going to apologize. I usually have two monitors, and apparently screen sharing with Max is a bit weird. So I have to share part of a screen and it might jump around here for a second, but I will do that. Share screen. Alrighty. Can everyone see that okay? Perfect. All right. So again, hi everyone. Thank you so much for having me here today and allowing me to speak with you. Today, I will be talking about a new genomics-based approach we have developed to monitor polar bears non-invasively. So before I start today, I want to take a minute to acknowledge that my project is part of a much larger project called BearWatch. And BearWatch would not be in existence and would not be possible without the partnership of Northern communities. I actually just got back a few days ago from the community of Joe Haven Univit, with whom I had the privilege to speak and talk about these tools that we've developed here. And I wouldn't be here to talk and I wouldn't be able to do the work I do without Northern communities pushing for non-invasive monitoring methods, without their sample collection, and without the unique perspectives and inputs they provide into the project. So with that said, I want to give a little bit of context. I think everyone is pretty aware that human-driven climate change is causing significant changes to the world's ecosystems, and Arctic wildlife in particular are being impacted by climate change and the associated rapid sea ice loss. And we study polar bears primarily for two reasons. One, because polar bears have both cultural and socioeconomic importance for Northern communities, but also because two, polar bears are at the top of the food chain and they have vast ranges that include both marine and terrestrial environments. Therefore, we can learn a lot about total Arctic ecosystem health just by studying them. However, we're missing much of the information needed to actually understand how polar bear populations are doing. Current monitoring is based primarily on surveys of the 19 subpopulations or management units, of which Canada houses about 2/3. These have been designated largely based off of mark recapture, which is invasive, and aerial surveys. However, surveys for many of the management units are infrequent. They might happen once every 10 to 15 years. And many of these subpopulations are data deficient, which is shown in kind of the brownish gray here. Thus, it's uncertain whether current management units reflect contemporary genetic patterns, especially in the context of a rapidly warming Arctic. So just to reiterate a little bit, these subpopulations are data deficient because the current methods that are being used are quite time consuming, they're expensive, they're labor intensive. It can cost over $4,000 just for one biopsy dart for one bear. All current monitoring is invasive, and there's been repeated lack of consultation and collaboration with Northern communities. Therefore, we need a polar bear monitoring program or programs that are non-invasive, cost-efficient, inclusive, and community-based. And we hope that the project that I'm a part of, Bear Watch, will help contribute to these monitoring programs. So Bear Watch as a whole has two main goals. The first is to establish baseline population data against which we can compare back to and use to monitor bears. And then as part of that, we want to establish tools that we can actually use to monitor those bears and assess their response to climate change and other human-mediated environmental impacts. And again, this goal is, the second goal is to contribute to community-based monitoring plans. So the test that I'm going to talk about here today is a tool that we're offering as an option to communities, a tool that they can have available to them and can choose if and how to move forward with. This figure shows how we envision the monitoring program to work from the science side of things. So I can't really speak as much from the traditional knowledge side of things. I'm going to focus on the science, but how it works is community members collect Scat for which they receive remuneration. And then the Scat sample is sent to our lab where we take it and we distribute it to tons of different labs for different kinds of analyses. And we can tell a lot about an individual bear from its Scat. We can look at what kinds of things it's eating. So its diet, we can look at microplastics and how those have built up in the ecosystem. We can look at environmental contaminants. However, understanding all of this information depends on being able to reliably identify bear based on their genetics. That is, being able to tell them apart and also being able to identify sex. So this is where my project comes in. And I'm going to ask, how could we use genetics and how can we use Scat to study polar bears? Traditionally, muscle tissue from harvest samples or biopsy samples have been used to do genetic studies on bears. So I'm going to briefly go over that. So when we would receive a muscle tissue or biopsy sample, we're able to pull out or extract polar bear DNA from it. And then we can use different kinds of techniques to look more closely at the DNA. And I'm just going to give a bit of a broad overview because I know we have a diverse audience here today. So to understand how we use genetics to study bears, we need to know that DNA is made up of 4 building blocks. Here, I represent them using different colors. So what we can do is we can zoom into one spot in the DNA. And at this spot, we can ask, is there a blue block? Is there an orange? Is there a pink? Is there a gray? And once we've done that at one spot, we can do it at another. And we can do it at another. And we can do it over and over and over again many times. So we call these locations in the DNA SNPs, or single nucleotide polymorphisms. There are locations in the DNA where these building blocks could differ. So there might be a blue building block or there might be an orange building block. And this happens thousands of times across the genome. And what we can do is we can repeat this question over and over of what building block do we see here or here or here, and we can start to see differences between bears. So if we pretend that these lines are DNA, we can see that bear number 1 has a blue block at one location, an orange at another, and a pink at another. If we look at bear number 2, we can see it's very similar at the first 2 spots, but differs at the third. If that pattern holds true across many, many spots, not just at 3, we can say that these bears are pretty similar and that they might be related. So they might be parent offspring or siblings. Whereas if we look at the third bear, it has completely different genetic building blocks or completely different markers, and we could say that it's unrelated. And we can compare these patterns of building blocks across many, many spots in the DNA to a barcode. Each bear has a unique genetic barcode, similar to like you would find at the store, but the barcode is in its DNA, and it distinguishes it from other bears. So again, this has been traditionally done with tissue or biopsy samples. And here, each bear that has a different genetic identity or barcode, I have just made a different color. The yellow bear has a different barcode from the red bear, for example, and that's different from the blue, which is different from the green. We have 2 green bears here. They're not quite the same color. So I would say that those are similar barcodes and could, again, be parents or parent siblings or parent offspring or siblings. However, we can do this without handling bears at all, just by collecting Scat. So if we collect Scat from a red bear, we can identify it as a red bear. We can get that same genetic barcode. And we can do that for the blue bear and the yellow bear. And we can tell that the 2 green bears are related. but how can we do this? When a scat sample arrives in the lab, the first thing we do is we extract DNA from it, similar to how we would with the muscle samples. The process is a bit different, but the concept is the same. Then we put the DNA into a test we call qPCR, and that can tell us how much polar bear DNA is actually in it. So if you recall, polar bear DNA has DNA from a lot of different, polar bear samples have DNA from a lot of different sources. It's going to have DNA from its diet. It's going to have it from bacteria. We just want to look at the polar bear DNA and quantify how much there is. And once we've done this, if we have enough polar bear DNA, we can then put those samples into a new method that we have developed and optimized throughout my undergrad and my master's work to genetically identify bears. This method is called genotyping in thousands by sequencing, or GT-seq. And overall, GT-seq allows us to identify those bears based off of those genetic barcodes. So what kinds of things can we use GT-seq data for? For local communities, this technique means that we can tell whether individual bears are returning each year to the community. We can also use Scat as a form of genetic marker recapture. So if these are the Scat samples collected in 2022, we can identify individual bears. Again, we'd look at the color here, and the red would mean that we've collected 3 different samples from a bear with the same genetic barcode. So we've collected 3 samples from the red bear and can tie it back to the red bear. Then, if these are the samples collected in 2024, some portion of the samples collected will be from bears that were already sampled in previous years, such as the red bear, and these are called recaptures. Some portion, however, will be bears that we've never gotten samples from before, and we can use these relative portions to get an estimate for population sizes in different areas. We can also use Scat to study movement or dispersal. So using biopsy or by collaring bears and following them, we could see in the past that the blue is different from the green and that the green bear has moved from one location to another. However, instead of doing that, we can just collect bear Scat and get the same information. We've collected 2 different Scat samples at different locations, and we can tell that the bear has moved. So some exciting news, we just received word that the paper we wrote demonstrating the successful use of this method for non-invasively collected Scat samples has been accepted for publication. I won't give you all the technical details on this method, but I will dive a bit into some of the results that we've gotten so far from using it. So as I mentioned, we used a test called qPCR to screen for samples that actually contain that polar bear DNA to start with. And we find that this is approximately 50% of the samples, which isn't a lot, but we did find it is possible to go back and re-extract important samples and go back and get that DNA if needed. It can be pretty heterogeneous across the sample, so we can go back, re-extract that DNA and try again. We find that using GT-seq, we can identify a bear and also assess population structure using only 324 genetic markers, or 324 of those building blocks. We actually need quite a smaller amount to identify the bear, but we need more to look at things like population structure. For non-invasively collected Scat determined to have polar bear DNA, we're able to obtain a clear genetic identity for 85.7% of samples. And we also show that we can identify the sex of bears based off of these Scat samples, with hunter-provided sex and GT-seq determined sex matching for more than 90% of the samples. Now I'm going to get a little bit more into some of the analyses we performed. And I'm going to say off the bat, this graph looks more complicated than it is. This is just showing probability of identity, which is the probability of accidentally identifying 2 bears that are actually different bears as the same bear. So all you need to know is that a low probability is good. We can identify a bear with high accuracy. And what this graph shows is that it quickly slopes down. And all this means is that we can accurately identify an individual bear with very, very few of those genetic markers or building blocks, really as few as maybe 30. But again, we use more so we can look at things like population structure. So using 324, we can be pretty confident that we can accurately identify a bear. This again looks more complicated than it is, but it just reiterates the idea that we can differentiate individuals. So we used our data to calculate relatedness values for each of the bear pairings in our data. And we wanted to look at how well we could differentiate bears that are either unrelated, they're full siblings, they're half siblings, they're parent offspring. And here we can see marked separation between those categories. And really, again, all this is showing is that we can reliably differentiate bears that are any level of relatedness. As I mentioned, once we identify bears, we can look at tons of things, including their population structure. So we look at population structure by considering how similar bears are. The more similar, the more likely they are to be put into the same genetic cluster. So bears here that are in the pink cluster are more similar to other pink bears than they are to those in the yellow cluster. And using Scat and hunter-collected samples, we found 4 genetic clusters in our data, 3 of which are very well known. The 3 previously studied clusters have been referred to as the Arctic archipelago, shown in yellow, the Hudson complex, shown in this bluish purple, and the polar basin, which is shown in pink. And the main takeaway from this that I want to emphasize is that what this is showing is that we're able to discern bears at similar certainty to and resolve similar patterns of population structure that were found using studies just with muscle biopsy, more invasive methods. So we're getting very similar results. However, there's important benefits to using non-invasive fecal surveys. We're able to monitor bears at a much greater cost efficiency than more invasive methods. We're able to collect tons and tons of samples more quickly and more cheaply. This data, importantly, is non-invasive. You're not interacting with the bear in any way. And there's more potential to directly involve communities throughout the monitoring process. Thus, GT-seq represents an opportunity for efficient assessment of Canada's polar bear populations. We can get the same information much quicker in a much more inclusive way. And as we continue sample collection at a quicker rate than the traditional methods, we can continuously update our understanding of population structure in the context of a rapidly warming Arctic. So just to finish off, our hope is that GT-seq will become part of community-led monitoring programs that will hopefully improve monitoring of Canada's polar bear populations, increase knowledge, and inform management efforts. Importantly, we hope that these programs will be a collaboration between traditional ways of knowing and Western science. And I want to mention that GT-seq is not specific to Scat or to polar bears. We have just found out that we can use it with bear tracks, which is very exciting, and it can be applied to other species. So hopefully we can set an example with polar bear monitoring, and maybe this approach can be applied to other species and act as a model for wildlife monitoring across the world. And I'd just like to take a moment to say thank you, and that Bear Watch would not be possible, again, without the support and collaboration of the new 𝐉𝐿𝐼𝐃 Council, Joe Haven and Coral Harbor Hunters and Trappers Association, Canadian Rangers, and all the Northwest Territories and Nunavut communities participating in the project. Thank you as well to our funding partners and all the other labs participating in Bear Watch, and all my colleagues within my lab as well. Thank you.
Okay, thanks, Kristen, for your presentation. It's really very interesting topic, and I like this picture. The map. Yeah, it's very nice. Anyway, I will pass it on to judges for panel discussion.
I can go first, and maybe we can do it like last session. So we take one question, Claris. Is okay?
Sounds good.
Okay, so my first question is, I'm really excited about your result. You truly conveyed the significance of this method. So my question is, can this method be used with other bears, with other animals, for example?
Yeah, 100%. So the thing with GT-seq is that you develop species-specific primers. So the methodology will be the same. You just need to develop those primers for your species of interest. We've actually just tested, we're still getting the results for it, but because grizzlies and polar bears are so closely related, we found that our qPCR test works pretty well with grizzlies, and we're testing it with GT-seq as well.
Excellent, thank you.
Yeah, okay. So I guess my question would be on the methodology, especially with the community side of it. I feel like definitely a huge part of the Bear Watch success will definitely be with the community involvement in it. I'm just wondering if it's also part of the program that you kind of, train and empower the local community. Because I know, like, for instance, sample collection, there's certain specific way to collect sample to make sure DNA is preserved. So yeah, can you tell me more about that?
Yeah, so I can kind of speak to a little bit of what I know on it, but more on the social science side of things. That we have other colleagues that focus on it, but I got to see a little bit more of the community engagement on my recent trip. And I know that different members of the team have been working with community members on, properly collecting samples, specifically, with snow tracks of, you have to be careful not to collect from multiple bears. You make sure it's one bear, you have a control. So that is definitely something that is in the works. And the goal of this is to provide tools back to those communities. And ultimately, the goal is to have that be something that they can use themselves and work for it in the future. So it's hard because, I'm on the science side of things, and you don't want to make, promises and statements that you can't necessarily hold up to. But the goal is to, supply these tools to the community and hopefully train them on how to use them so that they can use them themselves.
Yeah. Okay, awesome. Thanks so much, Kristen.
Yeah.
My next question is a bit more about the genomic method. You've worked in this for a while, so you started from other methods and previous work and stuff. Can you tell me a bit more about how this was developed and what are you building on?
Yeah, so kind of how I started out in the project was we hadn't settled on using GT-seq. There's a few different methods out there that we're looking at using degraded samples or fecal samples in some way. I think we considered the fluid gym system as well. So I did a really extensive literature review, and just kind of based off the methodology, GT-seq seemed really promising and very straightforward as well. There's a full pipeline posted online by the creator, Nate Campbell, and he is also very available to help get it up and running. We also had a colleague that had a connection to another lab that was also kind of working on GT-seq, and it just seemed like that was the way to go, to give it a try at least. So that was the first thing we tried. It showed a lot of promise. And then, so my undergrad thesis focused on validating it really before we got more into validating it on the lab side of things. So we went through and we created all these primers, and then we went through and we tested them. And we wanted to show that we could actually get enough genetic information to identify a bear and to look at things like population structure with a smaller amount of SNPs as we could a larger amount, because traditionally you're using thousands and thousands of markers. And so what I did is I used a data set that had our smaller number of markers. So now it's 324. And then I looked at a data set that had all of our available markers that were of sufficient quality. And I compared them on, things like, I looked at probability of identity, and it was pretty clear that we could get accurate genetic identity with very few. But then I looked at things like DAPCs and structure analysis and found that those results were super comparable between the two data sets, which told us that we could use that small number of markers to get similar insights. And then we moved forward with actually testing the GT-seq test itself, I guess. So we did this in a lot of different ways. My paper is very, very, very methods heavy, but I guess to dive into it a little bit, we did different tests with the primers. So we ruled out primers that were having, primer dimer effects, non-specific effects throughout the laboratory process, and we optimized the laboratory protocols themselves. And then as well, we had different kinds of samples. So really we had samples that were from harvest, just straight from quota. And then we had biopsy samples throughout the years, and we had the field collected feces. But then importantly, we also had these sets of, I guess, different bear parts that came down from harvest as well. So those sets contained muscle, and they contained liver and fat, and they contained intestines, which typically would house feces. So we use this kind of like a validation. We would have muscle be our gold standard of, this is probably the highest genotyping success, and this is what the actual genotype is. And we could compare the genotype that we got with the colon feces to the muscle to get an idea of genotyping success. We can acknowledge that that's going to be a lot fresher and is going to likely be better than the field collected feces, but it does give us a bit of a proxy as well. And then I also wanted to look at whether that qPCR step was useful at all as well. So we went through and we blindly put ahead a bunch of samples without testing it with the qPCR first, and then went back retroactively to see, okay, the samples that didn't successfully genotype, was there any polar bear DNA in them at all? Is this useful? So those are a few different ways that we looked at validating it. And then again, as I talked about in the talk, just looking at the results that we got and comparing that back to previous literature and showing that we get those similar results using these samples. Sorry, I know that was a really long answer.
But it was a long process, so you're honoring
Yeah.
all the effort it took. So thank you.
Yeah, I think you touched my next question a bit, but I just want to know, like, what are the challenges in, using specifically Scat samples to extract DNA, and how is your methodology adapted to address those like challenges?
Yeah, so that's a really good question. There's definitely a lot of challenges associated with using Scat. There's also, a really big push for it right now because it is non-invasive. The big thing is that the DNA in Scat is typically of very low quality and low quantity relative to muscle samples that have been traditionally used. And again, you get all that cross-contamination. So you're getting DNA from bacteria, you're getting it from diet, from all these different things that are in the Scat. Most of the DNA that is in the Scat is not polar bear DNA. A relatively small amount is, and that's just from the intestinal lining of the bear itself. So that's definitely a challenge. And then traditional methods of genotyping really require high quality DNA. So we originally tested this, I guess, Scat DNA with dd-rad, which is traditionally used for genotyping. And it just wasn't of sufficient quality or quantity. And then you can get things like different biases within your ultimate analyses because you're having it different things differentially genotyped, and it gets very complicated and biased. So using GT-seq itself was a method to work with the degraded DNA. GT-seq was originally published in 2015 by Nate Campbell, and he was working with degraded samples, which gave us hope that it would work with feces, which is very degraded. And then in the lab, we looked at modifying those protocols using not qPCR, but GT-seq itself is a multiplex PCR approach. So you're sticking a bunch of things in one tube and then amplifying them over and over and over again. And we played around with how many PCR cycles to use and how much to dilute original DNA samples, and tested that a bunch of different ways in a bunch of different combinations, and then found what seemed to work the best. And in terms of extracting the DNA, we also looked at grabbing it from different parts of the Scat sample itself. So where we actually subsampled it, I include some like subsample duplicates in the paper and compare the genotypes we get from them. So a lot of the time they seem to line up, but we have one sample where we got no successful genotyping. And so we considered successful genotyping. It was genotyped at more than 50% of the markers, but we got no successful genotyping from one subsample, and then the other subsample from the same Scat got near 100% genotype, like across all loci. So it does show that you can go to the different parts of the scat sample. So even doing duplicate subsamples when you're extracting and then maybe combining them could be helpful. We tested a few different extraction protocols and ultimately found that the quickest and most efficient seemed to be the 𝐒𝐏𝐃𝐄𝐑, but we're definitely open to trying different things there as well.
Yeah, awesome. That's a really good explanation. And I like how you make it like a bit laymanized. And so I'm able to like understand your explanation. Awesome. Thanks, Kristen.
Yeah, thank you.
I wonder, Noeen, if we have time for one more question. I see two really good questions from the audience.
Yeah.
Do we have time for one of my questions?
I think we have time, so you can go ahead. We still have lots of time actually. So you can ask as many questions as you want. We only have 2 or 3 questions from audience, not more.
Perfect. So my question is for your project as a whole, what are the areas of opportunity that you would like to carry on to continue working in, perhaps improving or just dwelling in?
Oh, geez, that's a good question. Yeah. Well, the one thing with GT-seq is that it can be modified to ask different kinds of questions, which is really exciting. So we optimized the panel for, looking at identifying individuals and sex and population structure, but it can be adapted to differentiate species. It can be adapted to look at, evolution in real time, things like outliers. So really, you can go back and you can modify what markers you include in your panels to ask different kinds of questions, which would be really interesting to go back and do. Yeah, so that's one thing. I guess in terms of, I'll clarify in your question a little bit. So kind of how I would like to move forward with the project in the future. Yeah, I think what this recent trip has taught me is I am really excited on seeing this get carried through and really interested in those conversations that are happening between scientists and communities. So it's hard because I am on the science side of things, like I'm a geneticist, but I think I would like to continue, communicating the results and the kinds of things that we can do, and also help with, training communities on how to use them. So that's kind of how I'd like to move forward with the project. And yeah.
Excellent. Thank you very much.
Yeah, I guess I want to just follow up on also that question. So I know you mentioned like another non-invasive way to track polar bears would be like through their tracks. I'm wondering if like in the long term, that is also something that will be part of Bear Watch and what you do there.
Yeah, so that's a big thing is we have a community that brought up that in their area, they don't really see a lot of scat samples, but they do see a lot of tracks. So having that as an option is really important. Obviously, with tracks, you're not going to get the same amount of information. You're not going to get diet. You're not going to get microplastics. But it is very helpful if you can identify a bear. You can look at dispersal. You can still look at things like population structure. So that is a goal. Like I think I just got an email today about we received a lot of raw sequencing data back from tracks, and we'll be looking at that. But the goal is to enable community monitoring through Scat and through tracks. Both ways, you don't have to interact with the bear at all. And GT-seq looks like it successfully works for both of those things, because with the tracks, you're looking at eDNA. So similar to water, like you collect a sample, and then there's environmental DNA. So there's skin that's been shed from the bear's paw, and that's going to be in really, really low quantities, but you can extract that. And because it'll be of lower quality and quantity like Scat, we can use GT-seq, and it's the same species. We're asking similar kinds of questions. So that ultimately will be a goal.
Yeah, awesome. Thanks so much.
And Noeen, if you want to proceed with the audience questions, I can save mine for later.
Okay, thanks, Cecilia. Thanks, Christine, for the answers. So I will start from Isaac's first question. So his question was, if you would elaborate on the development of your techniques in general, which I think you already did, so I will skip it and I will go to the next question. Next question is, if you could speak on how you develop relationships with communities such as 𝐒𝐿𝐄𝐄𝐄𝐃, I guess,
Haven.
𝐒𝐿𝐄𝐃𝐄𝐄, right? So 𝐒𝐿𝐄𝐃, how your work benefits these communities, as well as how your work has been improved by the inclusion of traditional ecological knowledge.
Yeah, so those are some big questions. I guess to answer the first one, how we develop relationships with the communities. So we have a colleague that's worked with these communities for a very long time. I would estimate 15, 20 years. So has very close ties to the communities. I'm not sure how that originally went about, but I know that he's very close and regularly goes up and visits, and they really respect his input and his insights. So that's kind of how I think Bear Watch had an in, is through our colleague. And then, going up and reporting back results. I think there's been a really big issue in the past with fly in, fly out scientists. They just retrieve the information that they think is helpful, and then you never hear from them again. And I know Steve has done a lot of reporting back to communities and to governments. And I think it's just important to have that engagement throughout the process, so that you're not just checking in at the start, checking in at the end. It's actual community engagement throughout. So hopefully that answers the first part. And so as well as how work has been improved with the inclusion of traditional ecological knowledge. Yeah, so that is another big question. I'm not sure I can answer this to the best, or the best of anyone could on the project, because again, I'm not more on the social side of things. But yeah, I mean, this project again wouldn't be possible and wouldn't be here without communities asking for it and pushing for it. They've been asking years for non-invasive monitoring tools. And if I go out on the landscape in a helicopter and try to count bears, I am going to have no idea where to look. Whereas community members, they're out on the land, and that is part of their lifestyle. They have a relationship to the land and can go out. They're going to know where, to find those tracks, where to find those scat samples, where to find the bears. So that's really, really important. And I've talked a lot with Saskia DeWilt, who is also on the project. She's more on the social science side of things. And my project is pretty genetics heavy. It's the development of this tool. But for Bear Watch as a whole, I think the goal is to work with communities together to create a monitoring approach. So it's not just, us coming in as scientists and saying, this is how you should move forward, this is how you do things. It's actually sitting down and having those conversations and asking questions back and forth. Even just talking with communities, the terminology polar bear management isn't the preferred terminology. It's coexistence or cohabitating with bears. So there's just, I think, lots of ways the project as a whole has improved. And I don't even know if I would say improved, but it's part of the project. It wouldn't be here without it.
Okay, thanks, Kristen. I think the next question is very much related to this question that you just answered. The question is, if you were able to utilize traditional community knowledge for developing on any of these tests, and I think you already answered a lot of it, right? So I will skip it. I will go to the next question because Cecilia also has one question that we can go to later on. So the next question is, if you could explain what you mean by snow tracks. Okay, I would like to know also.
Yeah, snow tracks. So I think if I go back a couple slides here, this. So when a bear is walking out on the snow, it's leaving its footprints, and community members, when they go out on the land and they see these tracks, what they can do is they can collect a sample of that footprint. They can collect a certain amount of snow, and within that snow, there's the DNA that has scraped off of the bear. So I guess I would compare this back to water again. So traditionally, eDNA is you might scoop up a soil sample, you might scoop up water, and within that, there's DNA from all the things in the environment that maybe pass through it that are excreting, gross bodily fluids, whatever. So if you scoop a bunch of water up, you're going to get a ton of fish DNA. If you scoop a bunch of snow up, you're going to get DNA from the bear that has walked across that snow. I wasn't involved in the eDNA extraction process because my work study focused mostly on the Scat, but it looks pretty complicated. There's some filtering, but overall, I think it's been pretty well optimized and works very well. eDNA is a really promising field. So yeah, you scoop up that snow DNA, snow sample, and then you'll want to grab a control sample as well, because you're going to see relative amounts of DNA between those two. So you would likely see, lower to no amounts of polar bear DNA in a snow sample, a certain number of feet to the right or the left, as you would with the main track. Yeah.
Okay, thanks, Kristen. And the next question is actually a fun one. What was your favorite field work experience? I would like to know that too.
My favorite field work experience. Oh, geez. I think this is hard because my project's mostly been like a lab-based project, but then I've recently gotten to go up to Nunavut. I think just, we got to go out with community members and go out and, look for seals and see how fishing works. And that was really cool. We got to go out, honestly, this is field work, but we got to go like ATVing through the Arctic tundra. That was really cool. We got a field truck completely stuck in the Arctic Ocean, going out to fish with community members, and then got the second truck that we brought to try to get the first truck out stuck in the sand. So honestly, experiences like that are just really funny. I feel like getting a field truck stuck is like a classic thing to happen. So honestly, just moments like that where, things go wrong and you have opportunities to talk to people and get to know the people you work with.
Thanks, Kristen. I will pass it on to Cecilia. You had a question, right? You can continue. We still have time. So,
well, just following up on your description of this field trip, is there anything you learned about how climate change is impacting these communities from their members?
Yeah. So I guess, yeah, a little bit here and there. Just speaking with community members, they've definitely noticed changes in recent years. So even while we were there, there was a spotting of whales, I guess, in the area that they hadn't seen anything in the area for over 30 years. I think they said 38 years. And speaking with different community members, they're noticing bears in areas that they haven't noticed them in years and years. So there's definitely changes going on, and they've talked a lot about the ice-free season. The summer is lasting a lot longer, and overall they're seeing changes. Yeah.
Okay, thank you.
any other question, Cecilia or Claris? You had any question?
No, I'm thinking good. Yeah.
You're all good. Any other question from audience? Okay, if no, then I think we can close the session. I will share my screen. Kristen, you can stop sharing your screen. And let me go back and find out how to share. Where is my file actually? Oh, I think I closed my file. So give me 1 second. I need to reopen. Let me close this. Okay, thanks everyone for joining us today. Thanks, Claris. Thanks, Cecilia. And thanks, Kristen. So if you would like to follow up our upcoming symposium events or other events, please follow us on Eventbrite. We always had sometimes wonderful workshops and some seminars. You can check it on SCWIST Eventbrite or SCWIST website for events. So our next and final round or final session of Symposium, 15th session, is going to be on September 15th, the same time. If you would like, please come and join us again. Dilrakshi would be the speaker from University of Alberta, Canada. And if you would like to get more updates about Symposium, you can follow us on our website. And I will actually update this catalog by end of this week, so you will get to know more information. Also, we are going to have a mega finale where we will announce winners on September 22nd. So please mark your calendar. The time will be same from 12 o'clock to 2 p.m. PST, Pacific Standard Time. And it will be also recorded. You will get a chance to meet sponsors. You will get a chance to meet committee members, to meet judges, to meet our finalists, and maybe our one chief guest. So please come and join us on our mega finale. You can find that information on SCWIST Eventbrite or on SCWIST events page. With that note, I will stop sharing my screen, and I will give my, I think, because I am here, so I will give my closing remarks first before I go to everyone. Kristen, it was really a wonderful presentation, really amazing work. And when I posted it on LinkedIn, as I told you yesterday, one of my colleagues, he is a director of some company in US, and he was really interested, and he actually asked me this question. I'm really interested in this presentation, but I cannot come. I am interested the work that she is doing for polar bears, and I think you mentioned that you like climbing mountains, so he was in which type of the kiddo is this, right? So thanks a lot for participating in Symposium and joining us today. And with that note, I will pass it on to judges Claris and Cecilia for giving your closing remarks, and then to Kristen.
I just want to thank you, Kristen, and everyone in your team for sharing your expertise, your knowledge, and for continuing caring and for our fellow beings in this planet, and for working in collaboration with our indigenous communities in Canada. So thank you. It was a great presentation and a great QA session.
Yeah, totally. Yeah, thank you so much, Kristen, for that very great presentation that you have. It's so easy to digest, and it's also great to know how, the Northern communities is working with scientists like yourself to make this, this Bear Watch project work. I think, as Cecilia mentioned, it's like with global warming, most of the time, for myself, we all mostly think of like the human aspect of things, like climate change, migration, and a human perspective, but there's also like wildlife that will be heavily impacted. So it's really amazing how science is working to make that happen and make wildlife management happen. So yeah, great presentation. Thank you so much for sharing your research and your work, and thanks to everyone who's able to join us.
I guess I'll save my thank yous. Yeah, thank you so much, for having me here today. It was a really, really great opportunity to be able to come and speak and, have a discussion with you guys. And I really, really appreciate it. I had a lot of fun. And thank you to everyone for coming and for asking questions. And I look forward to chatting with you in the future. Thank you.
Okay, thanks, Kristen. Stay connected and keep in touch. And thanks everyone for joining us today. Have a great day, great evening, great afternoon from wherever you are joining us. Take care and bye. I will close this session now.
Stop recording now?
I see Claris has gone, right? Let me stop recording first.